Okay, so you want to stay on top of the latest health breakthroughs, right? But without wading through tons of technical stuff, let's jump into something really fascinating today. The connection between our immune system and aging, specifically these things called senescent cells.
Right. Senescent cells. Think of them as cells that have basically just stopped dividing for good. We're not dead, but they're not growing either. And crucially, they start secreting this mix of
Yeah,
molecules.
this inflammatory mix, the SASP, I think it's called.
Exactly, the senescence-associated secretory phenotype, or SASP. Now, initially, this can actually be protective, like stopping potential tumor cells from growing, but
But over time, they hang around, they accumulate.
precisely, and that accumulation starts to cause problems. It drives chronic inflammation, tissue damage, basically contributing to a lot of age-related diseases.
So the big question is, how does the body normally deal with them? How does it clear them out?
Well, our immune system is supposed to handle that. We know certain cells like NK cells, natural killer cells, and macrophages, they're usually involved in clearing senescent cells.
Okay.
But the evidence suggests that this cleanup crew, well, it gets less efficient as we get older.
Which kind of sets the stage for this new research we're looking at today. It's
Yeah.
still early days, a preprint from Liu and colleagues.
2025, right?
That's the one. And it points towards a potentially different player from the immune system getting involved, a less common type of T cell.
The gamma delta T cells.
Exactly, gamma T cells. So our mission for this deep dive is to unpack this study, see what it might mean. But, big caveat here.
It's a preprint.
It's a preprint, so the findings are preliminary. Really important to keep that in mind. It's a first glimpse, not the final word.
Got it. Preliminary, but potentially exciting.
Mm-hmm.
So these gamma delta T cells, what makes them different from the regular T cells we hear more about? Well, they're pretty unique. You can almost think of them as bridging the gap between the two main arms of immunity, the fast-acting innate system and the more specific adaptive system. They have features of both.
Okay, so most people know about alpha beta T cells, right? The ones central to adaptive immunity.
Yeah.
How do these gamma delta ones differ in how they spot a problem cell?
Yeah, that's the key difference. Alpha beta T cells typically need other cells to present little fragments of antigens, like pieces of a virus or a tumor protein, using these molecules called MHC.
MHC, right? Major histocompatibility complex, like a formal introduction system.
Kind of, yeah. Gamma delta T cells often bypass that whole MHC system. They can directly recognize stress signals that pop up on the surface of cells that are, well, stressed. That includes things like infections, cancer, and importantly for our chat, senescence.
So they don't need the formal introduction. They can just sense the stress vibes directly, like spotting MECAB or ULBP, as you mentioned.
Exactly. MHC independent recognition, which is a really interesting feature.
That sounds, well, potentially very useful if you're trying to target something like senescent cells, which might be quite varied across different tissues, right?
But that's the idea. Plus, these GAL T cells are often found residing within tissues already. They're like local sentinels.
So they're already on the scene.
Often, yes. And they can respond quickly when they detect those stress signals. That quick reaction time is another potential advantage.
Okay, so let's dive into the Liu study itself then. They used mice looking at lung fibrosis.
Yes, exactly. They used a common model where they induce lung injury in mice using a drug called bleomycin. This causes senescent cells to build up in the lungs, leading to fibrosis or scarring. It mimics some aspects of age-related lung damage.
And what did they find these gamma delta T cells were doing in that model?
The really crucial finding was that the GODI T cells specifically recognized and killed senescent cells in the lungs. They identified these cells by looking for markers like P21, which is often high in senescent cells.
They killed them. How?
The main way seemed to be through releasing cytotoxic molecules, things called granzyme B and perforin. Perforin basically punches holes in the target cell membrane,
Okay.
and then granzyme B gets inside and triggers the cell to self-destruct. Apoptosis.
Wow. So targeted killing of the senescent lung cells? And did that actually help the mice? Did their lungs get better?
The results were quite positive in the mice, yeah. They saw less fibrosis, lower levels of inflammatory markers linked to senescence, and generally better lung function compared to control groups.
And importantly, any collateral damage,
Mm-hmm.
any signs of harming healthy cells.
According to the preprint, they didn't observe significant off-target toxicity or unwanted widespread immune activation, which is obviously a really crucial point if you're thinking therapeutically.
Definitely. Okay, so if, and it's still an if, these findings hold up and translate, what makes this approach potentially exciting for humans?
Well, think back to those unique features. The MHC independence could mean Therapies based on this might work for a wider range of people, potentially without needing complex tissue matching.
Right, like a more off-the-shelf possibility.
Potentially, yes. And their ability to recognize various stress signals might mean they could target senescent cells in different tissues, maybe involved in different diseases. Plus, that rapid killing action may be useful for acute situations.
You mean not just the slow, gradual buildup of senescence, but maybe situations where things flare up.
It's a possibility to consider, yeah. But again, we need to temper the excitement. There are significant hurdles between a mouse study and a human therapy.
Absolutely. Let's talk about those challenges. What are the big ones?
Well, first off, senescent cells themselves aren't all identical. They're quite heterogeneous. They can express different markers, release different SASP factors.
So a one-size-fits-all T-cell approach might not hit all the bad senescent cells.
It might be tricky, yeah. We might need more nuanced strategies. Then there's the whole issue of immune regulation. You need the therapy to be effective, but you absolutely don't want it to trigger autoimmunity or damage healthy tissue. Fine-tuning that balance is critical.
That tightrope walk of efficacy versus safety, always key in immunotherapy.
Always. And of course, the big one. Mice aren't humans. Our immune systems have differences, so what works in a mouse model needs very careful validation in human systems.
Right. And what about actually making this into a therapy?
Yeah.
Getting the cells, growing them, giving them to people, especially older people.
That's a huge practical challenge. Developing methods to isolate gay T cells, expand them reliably in the lab, potentially in large numbers, and then deliver them safely and effectively, especially, as you say, in older individuals whose immune systems might already be, well, aging.
Which brings us to immunosenescence, right? The aging of the immune system itself. How does that affect these gamma delta T cells?
That's a really important consideration. Research suggests that with age, the frequency of gay T cells can decline. Their function can change. Maybe they don't produce the same cytokines, or they don't proliferate as well when activated.
So the very cells we might want to use could be compromised by the aging process we're trying to fight.
It's a potential complication, yes. It might mean that simply taking an older person's gay T cells and giving them back might not be enough. We might need to, say, activate or modify them ex vivo outside the body before infusion.
Like giving them a boost in the lab, or maybe even engineering them somehow.
Those are definitely strategies being explored in the broader field of T cell therapy, and they could be relevant here. Another thing is the tissue environment. A diseased fibrotic lung, for instance, might be a hostile place for immune cells.
So just getting the cells there isn't enough. They need to survive and function in that environment.
Exactly. We might need ways to improve their homing to the target tissue and ensure they persist and stay active once they arrive. Lots of layers to this.
It really sounds like it. But despite the challenges, this gamma delta T cell angle does seem like a genuinely new avenue to explore for tackling senescence.
I think so. To sum it up, this Lu preprint gives us some pretty compelling, though early, evidence that Go T cells can act as senolytics agents that clear senescent cells in a living animal model. It opens the door to thinking about them as a potential basis for novel immunotherapies against age-related conditions.
And the key exciting bits are that MHC-independent targeting and the positive outcomes they saw in that lung fibrosis model.
Definitely promising aspects. But one last time, it's crucial we underline the preliminary nature. It's a preprint. Many questions remain. We need a lot more research, especially focused on human cells and systems, and on developing strategies that are both safe and effective.
Understood. A fascinating glimpse, but lots more work ahead. Which kind of leads to a final thought, doesn't it? We know senescence isn't purely bad. It has that tumor suppression role early on.
Right. That dual role is key.
So, looking forward, how could therapies, whether they involve gamma delta T cells or something else, be designed to selectively remove the harmful pro-aging senescent cells without messing up their beneficial functions, like preventing cancer? That seems like the ultimate challenge, wouldn't you say?
Absolutely. Precision targeting is the name of the game. It's a complex puzzle, and this research adds a very interesting potential new piece to consider.